A very good afternoon to all the learners. I welcome you all again to one of my sessions. This session will be on basics of plant tissue culture, and this unit actually belongs to your course, that is Economic Botany and Plant Biotechnology. So what we are going to study today is essentially some of the very basic principles or the techniques which are used in the plant tissue culture mechanism or plant tissue culture procedure. So we will first start our discussion with what is meant by tissue culture. Now, tissue and culture, that means if you take any plant tissue, it can be a somatic cell or it can be an embryonic cell, and if you culture it in any culture medium under aseptic condition or in the laboratory condition, because normally what happens is we grow the plants in the field, we grow the plants in soil or in the field condition, but it is possible that you can also culture the plants, okay, or you can culture any plant cell in a medium just like you cultured the bacteria in a standard medium. Similarly, you can culture any cell, plant cell, in a medium under laboratory condition, under sterilized condition, and then develop a plantlet or then develop a seedling, and from the seedling to the plant, and then you can transfer that plant into the field condition. And the entire process of tissue culture is also sometimes known as the micropropagation. So what is meant by tissue culture? You can see the process of raising the plants under in vitro aseptic condition. Aseptic condition means in a sterile condition. By culturing the explants. What do you mean by explants? Explant means any plant part. It can be a somatic cell, as I said, vegetative cell, or it can be any reproductive cell. And you culture them in tissue culture medium or in a nutrient medium. So there are different questions that comes in mind now. Which are the cells or which are the tissues or which are the organs that we are going to use? Number one. Number two is what would be the composition of the culture medium, that is the artificial or the synthetic medium where you want to grow the plants or the plant cells, and then finally the way how you transfer the plant and how they develop into a mature plant which you can bring in the field condition or which you can preliminarily grow in the greenhouse condition and then bring it in the field condition. So in that way, this entire procedure is known as the micropropagation or the tissue culture technique. So what are the important points which need to be followed? Number one is you need a proper plant part. Okay. Number two is you need to maintain a very sterile condition in the laboratory. Okay. That is, in these cases we normally need a tissue culture room, a separate tissue culture room where you can maintain the aseptic conditions. Okay. And then do all the tissue culture related works in that tissue culture room, and then develop a plantlet, and after it you can transfer it to the soil or in the field condition. Of course, you have to maintain the ideal condition like the supply of nutrients, proper pH, you need a proper temperature, you need sufficient amount of light. Okay, so these are some of the conditions which you need to maintain in the tissue culture room or in the preparation of the culture medium. So the entire process we call tissue culture or micropropagation. Now as you can see in this figure that you are growing the plants in the test tube, okay, which in the early days we used to call them test tube plants. Okay, just you must have heard about the test tube babies which are normally done through in vitro fertilization. So here you are actually growing these plants in the medium, okay, and you can see the plants growing in the culture tubes or in the bottles, and after the plants are fully grown you can transfer them to the soil, as you can see in the third figure. So these are all the plants which are actually generated through the tissue culture technique. Now what is the importance of tissue culture? Why we need to follow this, why this technique is so important in biotechnology? Okay, because tissue culture is actually one of the fundamental steps in plant biotechnology. because it has a significant role in crop improvement. It is useful in getting large-scale production of plants in a very short time because you don't have to depend on seasons. Okay, it is season independent. You are developing a condition in the laboratory and you can grow the plant in the laboratory, maintaining all the necessary conditions. Okay, so it becomes much faster to generate the plantlet. Any economically important plant you can grow through tissue culture and you can propagate it in a much faster time. Economically important plant species have been improved using the tissue culture technique. It has contributed to our understanding related to the factors responsible for growth metabolism. If you want to make any academic study, okay, that you can also follow during the tissue culture technique. You can follow all the steps, intermediate steps, okay, that is occurring during the development of a plant in tissue culture medium. Disease-free plant, nutritionally improved plant, okay, the salt-tolerant plant, environment stress-tolerant plants, pathogen-resistant plant. You can generate any such plants with greater benefits through tissue culture technique. Production of secondary metabolites. Okay, these are also, these can be produced. Secondary metabolites can be produced in bulk amounts through tissue culture technique. Now another very important point, there are many endangered species or threatened species or rare species. That means which are almost whose existence is at stake. So if you can propagate those plants in the laboratory condition, then you can maintain those plants. Okay, you can protect them to some extent from getting extinct or from getting endangered. Okay, so you can do it in a much faster time. So you can see that there are different utilities and this is followed for different groups of plants like cereals, grasses, legumes, vegetable crops. Okay, in any case you need to standardize the technique. So it is a very powerful and potential technique which is followed as a fundamental step to the plant biotechnology. Now if we go back to the history of this tissue culture technique, then this scientist Haberland, as you can see, he was the first to propose the importance of plant tissue and cell culture. In fact, he first actually tried to develop this idea that any plant can be grown, any plant cell can be grown in culture medium. See, there is a basic difference here because you cannot expect any animal cell. Okay, the beauty of the plant tissue culture is that unlike animal, where you cannot develop, you cannot just take any body cell and if you try to grow it in a culture condition, it will not develop into a full plantlet. Do you expect an earlobe, if you take earlobe and try to culture it in a nutrient medium, do you get a human? The answer is no. But the beauty with the plant system is that if you can take any somatic cell or vegetative cell, okay, from the plant body and try to grow it, then it will develop into a full plantlet. And this property is known as the totipotency, which I'll come shortly, that this property of generating a full plant from a single plant cell is termed as the totipotency. So every plant cell has this potentiality to develop into a new plant. Okay. So this concept was given by Haberland. He grew the tissue, certain plants like Eichhornia crassipes and then Pulmonaria mollissima. So he actually grew the tissues, he took the tissues from these plants and he developed a full grown plant on a synthetic medium which was provided with salt solution along with sucrose. And he also was the first to culture single palisade cells from the leaves. So Haberland is regarded as the father of plant tissue culture, and he also showed that artificial embryos can be successfully cultured. And from this actually came the concept of totipotency, as I said, where I mentioned that totipotency is actually the inherent potentiality of every plant cell to develop into a total plantlet, total plant or matured plant. So the potential of a plant cell to grow and develop into a whole new multicellular plant is described as cellular totipotency. It depends upon the inherent capacities of plant cells for differentiation. The capacity of a cell to get differentiated into other cell types is called totipotency. So this is a very, very unique property of any plant cell, and this term was given by Morgan in 1901. Later on, other scientists, they used this potentiality to develop different plants, okay, from different plant species by isolating certain plant cells and growing in the culture medium. Now, in this context, you should be familiar with two other terms, that is dedifferentiation and redifferentiation, which is a part and parcel of this tissue culture mechanism. So dedifferentiation, what is dedifferentiation? That is reverting of mature cells to the dividing state. See, you are taking any plant cell, okay, it can be root tip or it can be protoplast, it can be anther. So these are all mature tissues, but you are putting these mature tissues into a medium where the cells will again start dividing and will develop into a new plant. So this process is known as dedifferentiation, where any matured cell, when you put it in tissue culture media and you design the media in such a fashion that these cells will again revert back to the dividing state. The ability of a dedifferentiated cell to form a whole plant or plant organ is known as redifferentiation. So when you put them in the media, it's a dedifferentiation process, but once the dedifferentiation has started, then it will again have the ability to regenerate an entire plant, and this process is known as redifferentiation. So I hope you get the difference between the two, and these are actually connected with the totipotency potentiality. Now, differentiation of one organ directly from another organ is known as transdifferentiation. And to express totipotency, a differentiated cell first undergo dedifferentiation followed by redifferentiation. So, in any tissue culture process, there has to be these two processes. That is, first there should be a dedifferentiation, that is from matured cell to dividing cell, and then there would be a redifferentiation, that is the dividing cell finally again gives rise to a whole plant. That means again it gets converted into a matured plant. Now in India, the picture was that this plant tissue culture technique in India, it actually started at Delhi University, and the two persons, they are very renowned persons who actually they were from Delhi University, that is Panchanan Maheshwari, we call P Maheshwari, and SC Maheshwari. So they actually first showed the production of haploid plants under in vitro conditions, and some of us who are familiar with tissue culture technique and who have done our research works on this molecular biology and tissue culture, we know that how difficult it is to develop a haploid plant using the pollen grains or using the anther as the explant tissue. But they were the pioneers in doing that, and that is a very tedious task, and they should be given full credit for that. SC Maheshwari and Shipra Guho, they also developed these haploid plants from cultured anthers of Datura anoxia that opened the new area of androgenesis. And mind that this is a very, very difficult technique, that is developing a haploid plant, getting a haploid from anther or tissue culture, from anther or pollen culture, and they did it long way back, and they did it using, so the first technique that was standardized in India was the haploid culture technique or generation of haploid plants. I will come to anther culture or pollen culture in course of my further discussions. Now, techniques of in vitro culture of floral and seed parts were developed during this period. Endosperm culture and plantlet regeneration via organogenesis were achieved later. So each of these terms I will come what is meant by organogenesis. Now next I come to, supposing you want to develop a tissue culture laboratory. So what are the components that you need to keep, or what are the components that you need to purchase to develop a tissue culture laboratory? You need a laminar airflow cabinet. Just like in bacteria you need that. For microbiology work you need that. Here also you need a laminar airflow cabinet. You need an autoclave machine, that is for the sterilization of media, sterilization of the culture vessels, glass vats, plastic vats, all the instruments like forceps, needles. Okay, so you need to sterilize them. Culture room and or cabinets. shaker, culture room should be supplied with the ACs. Okay, you need to provide them with split ACs. Then shaker, that is especially we use them for the suspension culture, generator cell suspension, BOD incubator, and the glasswares and also the plastic wares. So you need Petri plates, okay, the culture tubes. So all these are the basic requirements which you need to first purchase before you need to start up your tissue culture work or develop a tissue culture laboratory. Next we come to what are the steps of plant tissue culture. Now first of all what you need is you need to select an explant. That means which plant part you are going to use for tissue culture technique. So you, as I said, you can use vegetative cell or somatic cell, you can use meristematic tissue, meristem, root meristem, shoot meristem, you can use that. Okay, you can use you can use protoplast, okay, you can use like leaf, root, shoot, anything. You can use any vegetative cell, somatic cell. Okay, you can even use embryo, which you call, in which case you call it an embryogenesis. Okay, so there are, you can use the anthers or the pollens, you can use the endosperm, you can use ovule. There are so many, both the reproductive tissue as well as the vegetative tissue or somatic tissue you can use. So you need to select the explant. Depending on what is your purpose, you need to select the explant or the plant part. Second point is you need to sterilize the plant part, because if you take any plant from outside field condition, it can be contaminated. So what you need, because you are doing it in tissue culture in a sterile condition, it has to be sterilized. So this sterilization is the surface sterilization. So for that there are different disinfectants, or there are different sterilizing agents. The most common of which is mercury chloride. Normally we use 0.1% mercury chloride, or you can use any hypochlorite solution, calcium hypochlorite. Even you can use alcohol, that is 70% alcohol. Okay, so these are some of the sterilizing agents, and people use different such agents. There is no hard and fast rule. Depending on the amount of contamination and depending on the purpose of your study or purpose of your work, you can come up with or you can use any of such sterilizing agents. So what you do is you immerse the tissue in the sterilizing agent for 10 seconds to 15 minutes. The time you have to actually standardize. That is, you might have to try, okay, you might have to standardize that what should be the time limit up to which I should go for the sterilization. Over sterilization is also not recommended, because sometimes it has an adverse effect on the tissue. Again, if you sterilize it for very short time, then also chances are that it might have contamination. It might be contaminated, or it might still have some contamination. So you have to standardize that. So once this sterilization is done, next what you have to do is you have to prepare the medium. Now regarding the preparation of the culture medium, I will come to that shortly, that you have different kinds of plant tissue culture medium. Now the choice is yours. Which of this medium are you going to use? So maybe one medium is good for one plant species, another medium is good for another plant species. So you again have to standardize that for my plant, which is the best medium where I can get the maximum micropropagation, or I can get the maximum number of plantlets, that you have to standardize. So a suitable culture medium is prepared depending upon the type of the explant to be cultured. The culture medium, so you prepare that, you have a composition, like which are the components that you need to add for the preparation of the culture medium, that you add, and then you transfer it, pour that culture medium into glass plates or in culture tubes, okay, and then you sterilize it. How will you sterilize it? You put it in the autoclave. You have the autoclave machine and you autoclave it for let's say 15 minutes. That is the standard procedure of autoclaving. We normally follow it here. Next is, once the media is prepared and once the media is sterilized and you have all these media in the culture vessels, that means either in Petri plates or in culture tubes, you have it ready. Next task is you need to inoculate. That is, you have already sterilized your plant tissue and now you need to put the plant tissue in the appropriate medium, that is sterilized medium, and that we call it as inoculation. Next step is incubation. That means now you have to incubate, that means now you have to allow the plant tissue to grow in the culture medium for a sufficient period of time under appropriate conditions of light, temperature and humidity, which you are actually maintaining in your tissue culture room. Okay, and then you need to subculture, because ultimately if you keep it for a very prolonged period in the same medium, the nutrients might get exhausted. So what happens is you need to subculture them, that is you need to prepare fresh medium, and then in a similar fashion you need to transfer the explant to the fresh medium till it grows, or till it tries to develop, or till it is capable of developing into a plant. And then finally, after you generate a full grown plant, you need to transfer it. We call it hardening. Hardening means from the tissue culture laboratory, you bring it outside in earthen pots, put them in soil, grow the tissue culture grown plants in soil, okay, and then finally it is ready. When it is, it is of sufficient strength, and when it has grown to sufficient level, then you transfer it to the field condition. So in that way, if you set up a tissue culture laboratory, then you can actually generate a large number of plants, and you can even commercially you can sell them. Supposing you have your own tissue culture farm, you can generate any economically grown plant faster, and then you can even make profit out of that. You can sell those plants, okay, and then you can earn also. So in that way, this is a very commercially, it is also a very feasible process, and commercially it is also very economically, and commercially also it is very helpful if we learn this technique properly. It will make you self-independent, Atmanirbhar, जिसको हम बोलते हैं. You will be like that only. So now we come to the media used for plant tissue culture. Various media have been tried for raising different cultures using different explants. A suitable culture medium is required for culturing cells, tissues. Now I am coming to the next part, that when you design a tissue culture medium, then what are the components that you need to? As I said, that different media have been actually designed, or it has been proposed by different scientists. But you have to remember that what are the principal components? Like you need to have some macro and micronutrients, you need to have a carbon source, organic supplements such as vitamins and amino acids, growth regulators, and a gelling agent. So these are the principal components of a plant tissue culture media. A complex nutritive medium composed of chemically defined compounds is called a synthetic medium. Now we will come to some of these media one by one. First is, this is the most popular medium, and most of the tissue culture laboratories, they actually grow the plants in this medium, that is Murashige and Skog medium. In abbreviated form we call it MS medium. This was developed in 1965, and a lot of laboratories who work on tissue culture, okay, they actually use this medium only. Now this medium was developed to induce organogenesis and regeneration of plants. Organogenesis, I am coming to it. Organogenesis means developing of the root and shoot, because you see, when you grow a plant, even if you germinate, if you place a seed, what will happen? The radicle and the plumule, they come out, they emerge. Like the radicle develops into root and the plumule actually develop into shoot. So similarly, when you put any plant part, it will try to develop root and shoot, because then only it will become a full grown plant. That is this bipolarity. Okay, that is one pole towards the root and one pole towards the shoot. So this will occur. So that is called as the organogenesis, and that organogenesis actually occurs in the medium. Now MS media consists of macro and micronutrients, a carbon source, reduced nitrogen, the B vitamins, and growth regulators. Another medium which you have is the Linsmeyer and Skog, which was developed in 1965. It has a similar component with some modifications of vitamins. used for inducing organogenesis, callus culture, cell suspension and micropropagation. Next, Gamborg, O.L. Gamborg in 1968, they developed another medium, Gamborg or B5 medium, which is a blend of nutrients like inorganic salts, vitamins and carbohydrates. But one of the unique feature of this medium is that it has a very high concentration of nitrate and potassium and a lower concentration of ammonia. So you have to see that maybe there are some plant species which will grow better in Gamborg media rather than Murashi-Jens-Cook medium. Because if you want to develop your own tissue culture farm, then you will also have to see that it is not only that the plants are growing, but it also depends how much plants you are generating in a shorter time. Because in industry, it is very important that the time and the number of plantlets that you are getting in a certain period of time, that is important. So you have to actually standardize the media. Which media will give you higher yield? That is important. So that is why all the importance of all these media cannot be undermined. Each of these media, it has its own utility. Okay, next, niche medium. It was developed later in 1969, and people use this media initially to grow the anthers. That is for anther culture of Nicotiana, that is mostly tobacco species. It contains a high concentration of thiamine, biotin and folic acid. So these are all the vitamins. This is a vitamin-rich medium. White's medium was developed in 1963. This media was developed for root culture. It has a lower salt concentration and a higher concentration of magnesium sulfate. N6 medium, it was developed by Chu and co-workers, and also they use this media for culturing anthers of rice plants. It promotes initiation, growth and differentiation of callus. Now I'll come to this, what is meant by a callus in tissue culture. Media named EI was developed specifically to culture soybean, red clover and other legumes. Haberland's media is used to produce whole plants from a single cell. So Haberland's media is also very, it's quite useful or important in tissue culture. Now, all these media, they have certain basic components. Of course, there may be some variations, like I discussed that some of the media, they have very high concentration of vitamins. Some of the media, they have certain high concentration of salts, particular salts. But all of them have some basic components. What are those? That is macronutrients, micronutrients. Macronutrients, nitrogen, phosphorus, potassium, calcium, magnesium, sulfur. And normally you have different salts of these elements, and the working concentration, the stock solution is always 10 times higher, okay, than the working medium. That is, you are preparing it at a concentrated form, but you need a much dilute form. Okay. Similarly, the micronutrients like iron, manganese, zinc, boron, copper and molybdenum, stock solutions are made at 100 times the concentration of the working medium. So you prepare the stock solutions, okay, and whenever you need them, you dilute them. Okay, from the stock solution you can always dilute it, and then you can prepare your culture media, but the stock solutions, you maintain it very carefully at cold condition, that is in refrigerated condition at 2 to 4 degrees centigrade. So of course you need a refrigerator in your laboratory. Amino acids are added to the media to stimulate the growth of cells, protoplast culture and establish cell lines. So you can apply the amino acids in different forms like casein hydrolysate, L-glutamine, L-glycine, L-arginine and L-cysteine. So these are very common forms of organic nitrogen which are there in your media. Sometimes you need some supplements, organic supplements, protein hydrolysates, coconut milk, yeast extract, malt extract, potato extract, tomato juice. It's not that you have to add all these components. Maybe any one of them, or sometimes you may not require them at all. Okay? It's not that you have to add mandatorily the organic supplements, but if you get a better yield, that you have to try actually. by adding any of these supplements, are you getting better yield? Are you getting better production? So that you have to try. In some media, activated charcoal is used, which is useful in reducing some toxic compounds produced during the culture and promotes the unhindered growth of cells. Very rarely you need to add some antibiotics like streptomycin and kanamycin, that is in very low concentration, so that there is no infection. Now this is very important, that when you are growing your tissue culture grown plants, even in under aseptic condition, there may be contamination. Okay, sometimes you find that your medium gets contaminated, like with bacteria or with fungus. This is a very, very common observation in your tissue culture experiments. And then your entire work comes to zero. You again have to start from the beginning because your culture media has got contaminated. So however careful you have done the sterilization part, somewhere there might have been some lacunae, or somewhere there might have been some sort of mistake or carelessness, so that what happens is, even after your initial phase, your explant is perfectly fine, but maybe 10 days, 15 days after, you get some contamination, fungus growing in your media, because this is a very nutrient rich media. It contains sugar, carbon, it contains sugar as the carbon source. So you see that there is a contact, and then your entire tissue becomes contaminated and your work, it comes to a failure, and then again you have to start from the beginning. So this is one of the major problems, and you have to be very careful when you are doing experiments with tissue culture. Especially in your future, if you pursue research as your career, okay, then you might have to face this problem, but again you need to be careful. You need to do each and every step very carefully so that there are no chances of contamination. You eliminate the contamination chance. Now also you need to add hormones. This is very important because particularly auxin and cytokinin, you need to maintain a proper balance of auxin and cytokinin in your medium. For instance, the basic plant physiology says that auxin promotes rooting and cytokinin promotes mostly the shooting. So a proper balance of auxin and cytokinin is required. Okay, so that there is both the root formation as well as the shoot formation. Sometimes if you want to induce too much of rooting, then you need to put a higher concentration of auxin. Similarly, if you want to induce callus formation, you might need to add a little bit higher amount of auxin in the form of 2,4-D, because auxin comes in different forms, as you all know. Some of the, if you have gone through plant physiology, then you might have seen that auxin, it can be available in IAA, indole acetic acid, indole butyric acid, naphthoxyacetic acid, 2,4-diphenhoxyacetic acid, 2,4,5-T, 2,4,5-triphenhoxyacetic acid. So there are different forms of auxin. And again, which form of auxin you need to add, that again you have to standardize. And that might vary with your species. Similarly for cytokinin, it might be kinetin, it might be zeatin, okay, or it might be any other form like BAP, that is benzylaminopurin. So there are so many forms. So again, it depends which cytokinin you are going to use and which is going to give best result for your experiment or for your plant species. So you see, tissue culture is more like it's a technique, and you have to actually develop that technique. You will have to standardize that technique on your own for an unknown plant species particularly. For known species it is okay, but if you want to standardize or grow any unknown plant species which nobody has grown before, then you might have to make different types of, there are so many variables, okay, and you might have to standardize all those variables. So proper balance of auxin and kinetin in the medium influences morphogenesis. High levels of auxin favor rooting, while low levels help in the formation of shoot, and intermediate levels to the proliferation of callus or wound parenchymatous tissue. So what is the concentration of auxin and cytokinin in the medium that I am going to use is a very, very crucial factor in tissue culture. Now, if you want to grow the tissue in a solid medium, I hope you understand what is meant by solid medium and liquid medium. Liquid medium means it's a culture, and solid medium means it is solidified medium. So how do you solidify the medium? Because you are preparing a solution containing all the salts, carbon source, that is sugar, okay, the vitamins, the amino acids, the hormones. But then how do you solidify the media? For solidification, you need to add agar. And this agar is normally used as a gelling agent. Generally 0.5 to 1.5% of agar is added to the medium. So you add agar at the end, and then you autoclave it or sterilize it, and then leave it for cooling. Automatically it will gel, or it will turn, the media will turn into a solidified form where you can grow your tissue. Sometimes iron source such as iron EDTA is also added to the media. So you see, these are all the basic components of a tissue culture medium. Now what will be the pH of this medium? pH is normally set between 5.4 to 5.8. It should not exceed 5.8. It can be in between. It can be even 5.7. No problem. So this pH is very, very crucial. You need to, when you are preparing a medium, make sure that after the preparation of the media, after adding all the components, you check the pH. So you have the pH meter. Okay, don't check it just by a pH paper. Okay, you check it. There is an instrument called pH meter which shows the pH in a digital form, and you check it in that instrument, and then you are ascertained that yes, my media has this much pH, and then you can proceed. Then you can add the agar, and then you can solidify the medium. So that's all about the basic components, that is the the preparation of the medium, components of the medium. Now we will go into what are the different forms of tissue culture, what are the different forms in which we actually see the tissue culture technique, or what are the different purposes of tissue culture. Now, as I said, that when you take any plant tissue, okay, you put it in the tissue culture media. So what happens is you try to develop a plantlet. What is meant by development of plantlet? Development means you try to develop the root and you try to develop the shoot. That is, these are the basic organs. Root and shoot are the basic organs of the plant. So you have to develop that root and shoot by putting the plant cell in a culture medium, and this process is called as organogenesis, because you are generating an organ. You are generating the entire plant with the root and the shoot system. So organogenesis is actually nothing but it's the regeneration of the entire plant from a single cell, or from an explant, or from a single plant cell. So what are the important factors? As I already said, that you need to add all the components, and you need to add the auxin and cytokinin in proper proportion, because the auxin-cytokinin ratio actually determines the process of organogenesis. If the auxin ratio is high, it will promote more rooting. If the cytokinin ratio is high, it will promote more shooting. So if you need both, then there should be a proper balance between, or a proper ratio should be maintained, so that you have both the rooting and the shooting process. So this is called as the organogenesis, and organogenesis can occur in two ways. One is direct organogenesis, that is from the cell you directly develop the root and shoot and the entire plantlet, and another one is indirect organogenesis, where you actually develop a callus. What is meant by callus? Now callus, as you can see in this figure in number A, that is actually a callus. So it is a mass of polyploid cells. Okay, it's an undifferentiated mass of cells. So you put the plant tissue in such a media that it will first develop into a callus, and then you grow the callus. So callus is an unorganized, a callus is a mass of randomly dividing cells, and a mass of polyploid cells, very unorganized, okay, and then it goes on proliferating, and then you transfer it in the regeneration medium where it will try to develop into the organs. So initially you have this callus, and then this is called as the callus induction medium, and then so callus induction medium will have more kind of certain auxins, like you can add 2,4-D, 2,4-dichlorophenoxyactic acid. Once the callus is formed, you transfer it to a regeneration medium where you shut off the auxin, or you lower the concentration of 2,4-D, and then you try to develop the organs, that is the root and shoot. So from callus, via callus, you are getting a plantlet. So this is called indirect organogenesis, and direct organogenesis means where you are not developing any callus. So this is the basic difference between the two organogenesis process. Now organogenesis has been obtained from the culture of various explants such as cotyledons, hypocotyl, stem leaf, shoot apex, and again organogenesis will again involve two steps, as I already discussed, that is dedifferentiation and redifferentiation, which is also true here. Now whenever there is a root formation, we call it rhizogenesis, and when there is a shoot formation, we call it the collagenesis. So rhizogenesis and collagenesis both are very common for the organogenesis process, because ultimately you need both to generate a plantlet. You need both root as well as the shoot, and you are developing that in the medium. Now naturally this will depend on various factors, chemical factors and physical factors, that is the physiological state and size of the explant. Of course the explant that you are going to use should be a healthy tissue. The environmental condition like temperature, quality and quantity of light. High light intensity has been shown to inhibit shoot bud formation. Blue light promotes shoot bud differentiation while red light stimulates rooting. Temperature up to 33 degrees centigrade supports growth of callus, but lower temperature stimulates differentiation of shoot bud. As I already said, you need to have a moderate to high concentration of auxin for callus formation. But if you don't need the callus, don't go for it. Don't add 2,4-D to such a high concentration. Okay? But if you want specifically the callus induction, then you prepare a medium which we call as the callus induction medium, where you have a very high concentration of 2,4-D. Now the next step, which is also very interesting, is the somatic embryogenesis. Now normally embryo, it actually develops from a zygote. You all know that. Zygote, it transforms into embryo. But the beauty in plants system is that you can develop embryo-like structure, which you call as the embryoids, from somatic cells, from vegetative cells, and these are called the somatic embryo or embryoids. Now why you need to induce the somatic embryogenesis? Now this somatic embryogenesis can also be either indirect or direct. Direct means when they develop directly into the entire plantlet, and indirect embryogenesis, that is when you again induce callus formation, and then you develop the you try to develop the plantlet out of it. Now the question is that why do we go for somatic embryogenesis? The most important part of doing somatic embryogenesis is the somatic embryos. Okay, you can use this somatic embryo as artificial seeds or synthetic seeds. That means these kind of seeds, normal seeds, they have a dormancy period. Okay, but here in case of somatic embryo or the artificial seeds, they do not face this dormancy. So it is faster. They can be just sown and you can generate a plant much faster because they don't undergo or they don't pass through the dormancy phases. But then these need to be properly packaged with nutrients. So you have the concept of artificial seeds where you can use this somatic embryo, or you can package these somatic embryos with certain nutrients, encapsulate them, and then make them behave as seeds, or grow them or sow them in soil condition, overcoming the dormancy period, so that you have a very, very faster regeneration, or you can get plantlets in a much faster time. And that is why we go for, this is the main purpose of doing somatic embryogenesis. So this was first done in carrot, Daucus carota, by Stewart and co-workers, and later on it has been done with other plant species as well, citrus, coffee, ziames, etc. So the process of somatic embryo has three main steps. Now see this figure, that if you compare a somatic embryo and a zygotic embryo, then in case of somatic embryo, okay, you have the callus, and then you have three phases of growth: globular stage, heart-shaped stage, and torpedo stage. You can see this, that is, this is the globular stage, then this is the heart-shaped stage, and this is the torpedo stage. But in case of zygotic embryo, you have another phase, that is the cotyledonary stage, which is not there in case of a somatic embryo. So you can bypass this stage if you go through somatic embryogenesis. So the development is faster, as I told you, because directly from the torpedo stage you have the bipolar embryo, and bipolar embryo means it has the root and shoot formation. Whereas in case of zygotic embryo, that is the embryo developed through that is the if from the zygote you develop the embryo, then you also have this cotyledonary stage. So it will be a little bit more time, but again it has a desiccation and dormancy phase, which is also not there, so that you can regenerate you can regenerate a plantlet throughout the year, whereas here there is a dormancy phase. So I hope you understand why we go for somatic embryogenesis, because it is faster. You can use this somatic embryo as a seed and propagate a plant much faster, okay, rather than using a normal seed which has been developed through embryogenesis from the zygote, okay. So somatic embryos act as valuable source for the large scale propagation of germplasm resources, and as I already said, you are generating artificial seeds. Now another technique of tissue culture is the protoplast fusion and somatic cell hybridization. What is meant by protoplast? Protoplast means when you take any cell and you remove the cell wall, okay, then the mass that remains left, that is called as the protoplast, that is minus the cell wall. So how do you do that? You can remove the cell wall either mechanically, in which case you call it a mechanical method, or you can do it through some enzymatic treatment, okay, that is you treat the cell with certain enzymes which will actually degrade the cell wall. So this process was first developed by Cocking in 1960, and actually he used certain enzymes to release the protoplast, to degrade the cell wall and release the protoplasts. So cellulase was isolated from the culture of fungus, Myrothecium verrucaria, to degrade the cell wall, and later on different other cell wall degrading enzymes have been used. So mechanical method is not that much common, because you just select some highly vacuolated cells, okay, the plasmolysed cells, withdrawal of contents to the cell center, and then you dissect the tissue and deplasmolyze to release the preformed protoplasts. But this is not a very routine method, because here the yield of the protoplast is very low, okay, and also because of the mechanical there can be mechanical injuries to the tissues. So we mostly use the enzymatic method, that is we treat the tissue with different kinds of cell wall degrading enzymes like cellulase, pectinase, okay, or the macerozymes in different concentration, hemicellulase, and usually we use the mesophyll tissue because here the cells are very loosely arranged, so that if you add the enzyme, then the enzymes will be better accessible because the cells are very loosely arranged. So you can use the different enzymes as I have listed here: cellulase, then the drusilase, hemicellulase, pectinase, pectolase. So again they come from the fungal sources mostly, okay, like trichoderma, then aspergillus, rhizopus, and you can use them at different concentrations. So this is actually the figure for protoplast, that is isolated protoplast. Now you can actually fuse the protoplasts using certain chemical agents which are known as the fusogens. Now one such very important chemical agent is known as the polyethylene glycol. Now why do you fuse protoplasts? To overcome the incompatibility barrier. Sometimes it is not possible to cross, okay, to artificially cross between two varieties or between two species, because there is an incompatibility barrier. You cannot generate a hybrid because of crossing. So in that case, the alternative approach would be to go for protoplast fusion. That is, you isolate the protoplasts from each of these two species and then try to fuse the protoplasts of the two species, so that you get a hybrid. Okay, and that hybrid you call as the somatic hybrid. Normally this is a very difficult process, but we can do that using certain chemical agents like sodium nitrate. You can use high pH, high calcium treatment, and last but not least, you can use the polyethylene glycol, that is 28 to 56%, which actually enhances the agglutination and fusion. It actually induces the fusion of the protoplasts. Okay, and there is certain mechanism, because actually PEG is negatively charged, and if you use calcium that bind to the negatively charged molecules in the plasma membrane, the PEG molecules can bind to the cationic molecules of the plasma membrane, and then high pH, high calcium, it neutralizes the negative charge and allows the protoplast to come together, to come in close proximity. High temperature also you need to add, or you need to subject them to high temperature, so that there is a perturbation of the lipid molecules in the plasma membrane, and all these together actually will initiate fusing of the two cell masses, fusing of the two protoplasts, and ultimately they will actually fuse and become one. Okay, of course there is a cytoplasmic mix as well as a nuclear mix, but mind that if there is a nuclear mix, then there is a chance of increase in the ploidy level. So what you do is you eliminate one of the nucleus, so that there will be only a cytoplasmic mix, and you call it a cybrid. So you take the two protoplasts from the two species, fuse them, and then generate a plant out of this fused protoplast. Of course, you need to identify and select whether the fusion is successful, and there are different techniques. You can use morphologically distinct cells, you can label the cells with different fluorescent dyes, and so on. Okay. Lastly, there is another technique, that is anther culture and pollen culture. That is, you take the anther or the pollen grains in this case, and you try to grow them in the tissue culture medium. Now why we are going for this technique? Because you know the pollen grains are haploid tissue, and sometimes we need to generate a haploid plant. Why? Because if you can generate a haploid plant, then you are actually generating a homozygous plant. For obtaining homozygous lines, the plants derived through anther culture are subjected to diploidization. That is, if you actually culture the anther, actually it is the pollen grain which is developing into a plantlet. But the plants which are generated from the pollen grains are haploid, so they need to be diploidized, and for that you need to treat them with colchicine, and then you get the homozygous plants. So that is actually the utility of the generating the haploid plant through the pollen culture or anther culture. So you see, we have gone through different techniques. We have gone through organogenesis, we have gone through protoplast fusion technique, we have gone through anther culture technique. So there are so many things. You can get callus formation also. But the ultimate aim of all these technique is to propagate a plant, to get a plant in a very shorter period of time, and through micropropagation you can generate a plant in a much faster time, and which is season independent, okay, which is independent of all the seasonal variations and all the other factors in the environment. Okay, so this is a very powerful technique, and also a very interesting technique. It's a very well-known laboratory technique also. Okay, and I hope you have enjoyed this session. Please go through the module, and if you have any difficulty or if you face any difficulty, please contact us, write to us, and we will be there to help you. So thank you all for listening to me, and have a very, very happy learning. Thank you.
Now I will discuss about sanitary and hygienic requirements for specific food establishments like fruits and vegetable processing industry, milk and milk processing industry,